So today's video is going to focus on the cell membrane and how that cell membrane allows for cell transport. So we'll be looking at different examples of cell transport as well. So the cell membrane has a variety of components. We're going to focus first on the main component of it, which would be these phospholipids that you see here, these blue ones with the black tails. We'll look at those more detail on the next slide. And we're going to mention here that it's called the fluid mosaic model. The cell membrane is structured using this fluid mosaic model, meaning essentially that the components of the cell membrane, that they are flexible, and not just flexible in the sense that like they can bend, that they can kind of scoot side to side a little bit. So it is not a static structure. Static meaning nothing changes, nothing moves. The cell membrane is a fluid structure, and it's a mosaic because it has a variety of components. So that's our fluid mosaic model that represents our cell membrane. So let's focus first on the phospholipid, since the phospholipid, it makes up the bulk of the cell membrane. So it is our main component of the cell membrane, and it's arranged in a bilayer. Bi means two, and you can see that being illustrated down here in this picture. So the phospholipid has some distinct components to it. The top part, or the head, the circular part there, contains a phosphate group, which is attracted to water. The tails here, these contain the fatty acids, which are made up of lipids, and the fatty acids are repelled by water because they are what is called nonpolar, where that phosphate is polar. So the phosphate group has a charge, and the nonpolar parts of the molecule have no charge. If you think about salad dressing, right, oil and vinegar salad dressings, they settle out in the bottle and you have to shake them because the water part of the salad dressing, the vinegar, doesn't blend with the oil part of the salad dressing. This is a key component of how your cell membrane is going to work to form a barrier between the outside of the cell and the inside of the cell. So these molecules, these phospholipids, will arrange themselves with the polar charged heads facing the outside and the inside of the cell, and then the middle part will contain these non-polar tails that will have the lipid or the fat part. It is a semi-permeable membrane. Permeable means that things can pass through it. Semi is half. So semi means that some things are able to pass through it, not everything. Smaller molecules that they can have a charge, but they have to be really small. They can't be a full-blown ion. They can only have a partial charge, or completely nonpolar molecules can pass through. Larger molecules or ones that are completely charged like an ion cannot, and we'll talk more about those later. So the main job of the cell membrane is to define the cell. So what this means up here by defining the cell is essentially form a barrier, separate the inside of the cell from the outside of the cell. If there is no separation from inside to out and things are allowed to just move back and forth however they want from between the cell and the outside surrounding fluid, then you're not maintaining that inside environment of the cell. And that has to be maintained for the cell to stay alive. There are certain constraints that have to be met. And if there's open transport between the outside world and the inside of the cell, we can't maintain that. So that's the main job of the cell membrane, is to help define the cell by acting as that barrier separating it. We've already talked about the phospholipid component. So that's the major component set up in that bilayer again, which really determines that what is able to go in and out. Let's look at a few of these other components. These little yellow ones right here, these are cholesterol components. Cholesterol is going to help the membrane remain fluid. Remember, we talked about on the very first slide that meant that the membrane would be able to retain its flexibility. So cholesterol is going to help the membrane remain fluid. That's really important when temperature fluctuations and such happen so that it doesn't become hard and firm. We've got a wide variety of proteins that are within the cell. Our focus mainly is going to be these ones right here with the hole in them. These are channel, sometimes called transport proteins. These proteins allow those molecules that are polar, that have a charge, ions, large charged molecules, to be able to easily cross the membrane, because you'll see how this protein shelters it from the nonpolar part of the phospholipid there. So it allows those larger charged molecules to easily cross the membrane. And then another one that we're going to look at here are these hexagon-looking ones here that are attached to the proteins, not the ones that are attached to the lipids, but the ones that are attached to the proteins. These are called glycoproteins. Glyco like glycogen, so that would be a carbohydrate or a sugar attached to this protein molecule here. These act as name tags for the cell. In a multicellular organism, it's really important that the cells can determine what the other kinds of cells around them are, and the glycoprotein acts as that ID or that like name tag so that the surrounding cells know what kinds of cells are present with them. So let's shift our focus a little bit now to how things move back and forth across the membrane. So our form of transport that we're going to talk about first is going to be diffusion or passive transport, a form of passive movement. So passive movement, meaning that this does not require any input of energy. So no ATP is required for these molecules to be able to move. It is a form of passive transport where things are moving from high concentrations to low concentrations. So if we look at this example here on this membrane, we've got a semi-permeable membrane here. We can see that it's semi-permeable because it has those membrane pores in it right here, these openings where things can get out. So this is a semi-permeable membrane. Any molecules, let's say we had some molecules in there that were this big, right? They wouldn't fit through those openings, so they wouldn't be allowed to pass through. So this semi-permeable membrane allows those little orange molecules of dye to move through. If we look on the left side here, this has a high concentration of the dye molecules, where over on the right, there's a very low, non-existent concentration of the dye molecules. So the dye molecules are going to want to naturally just spread out, and they're going to spread across this membrane. So they're going to have traveled across this membrane, right? And they're going to spread from a high concentration to a low concentration until they reach equilibrium. And that spread of molecules is not going to require any input of energy. Molecules that can do this across the cell membrane are things like gas molecules, like CO2 and oxygen. They can easily diffuse across the membrane. Small molecules, and again, uncharged molecules, while they can be polar, they can have a partial charge, like for instance, water. They can't be an ion, like a full charge, like for instance, a sodium ion cannot diffuse across the membrane. So small uncharged molecules can diffuse across the membrane. Okay, let's look at a more specific example of this with osmosis. Osmosis is the diffusion of water. So again, as soon as you hear diffusion, you need to be thinking no energy, high concentration to low concentration. That is going to be characteristic of diffusion regardless of what type it is. So osmosis is the diffusion of water. What'll happen a lot with a semi-permeable membrane is that water is able to pass through it, but the other molecules are not able to pass through it. And so we talk specifically about the movement of water. If we look at this picture on the left here, these kind of bluish looking molecules, those are water. Those molecules are able to fit across the cell membrane. So if we look at this image here, Water has a high concentration on the left. Yes, I understand these green molecules have a high concentration on the right, but what that means then is that water is at a low concentration on the right. So water is going to flow from high to low, trying to reach equilibrium. And eventually you'll have enough water will flow across the membrane so that even though the green molecules stayed, when I add more water to this, right side here, it is going to change the concentration of green molecules. Think about it like diluting Kool-Aid, right? If those green molecules were the sugar in the Kool-Aid, as I add more water, right, I dilute that concentration of sugar Kool-Aid in that drink. And eventually it will reach equilibrium. We see the same example happening over here on the right. We've added a little bit of vocabulary. So we're going to talk about these terms real quick. So you'll see I have two types of solution on each side. This is called a U-tube, literally a U-tube. Okay, so I have two different concentrations of solutions on either side of this U-tube. Okay, this is a time again where prefixes and suffixes will be important. Hypo means below or less. Hyper, like hyperactivity, right, means above or more. When we see tonic here, tonic is referring to the solute, which is the part of the solution that's getting dissolved or broken down. So examples, that would be like salt, sugar. Those are our two big examples of solute. Water would be the solvent. Okay, so if I have a hypotonic solution, that means I have a low solute solution. So just a little bit of sugar, just a little bit of salt. If I have a hypertonic solution, that means I have a high solute solution. If I have a low solute, then I have to have more water. You'll notice they're both at the same level here. So if I have high solute, then I have to have higher water. If I have low solute, I have higher water. If I have high solute, then I have to have lower water. You can see that from the image, right? I have a few of those solute molecules, of those maroon molecules here. So I have a low solute, which means I have a high water. Over here, I have a high solute, which means I have a low water. And these are the kinds of things you write out the first few times. And then after that, you're like, oh, okay, I got it. Okay. Water always flows high to low. So osmosis will occur. Water will move from the left side to the right. You'll notice the right side increased in volume, but now their concentrations are the same. And the solutions have reached equilibrium. That required no energy. It just required the water to flow from a high concentration of water to a low concentration of water. Okay, so we're going to add one more vocabulary term to this hypertonic and hypotonic, and that's going to be isotonic. Isotonic means the same. So this is when the two solutions are going to be at equilibrium. That's the point you're trying to get to, is to an isotonic solution. If you have isotonic solutions on either side of the membrane, so if I have isotonic solutions here, the water is still moving. It's just moving back and forth at the same rate. So I don't see any major changes in volume, how in the U-tube before we had the U-tube and they started at the same volume. And then when the water moved, they ended up at two very different volumes. In an isotonic solution, the water, and so at this point, the water would still be moving back and forth just at the same rate. So you don't see any changes in mass, any changes in volume, anything like you would if the two solutions were hyper and hypotonic compared to one another. One of the biggest things that confuses people with this, so we're going to emphasize it again, is that with these types of solution, we're always referring to the movement of water but the solution is talking about the solute concentration. So again, those hypertonic solutions have high solute, therefore they have low water, and a hypotonic solution is always going to have low solute, therefore it will have high water. Also realize that these terms are just comparison terms. There's no set concentration to make something be hypertonic or set concentration to make something be hypotonic. If you have a hypertonic solution, that means there has to be a hypotonic solution on the other side of the membrane. Again, these are just comparison terms. I could have a beaker. This is a common example. You see a beaker with a cell, and that beaker could be 97%. Let's do solute. It'd be super saturated. This would be a crazy amount of sugar or something to have in the solution, but let's do it. So 97% solute, and this cell could be 98% solute. Both are really high, but what that means for the cell, right, is that the cell would be 2% water. The beaker would be 3% water. Water flows high to low. The beaker is the hypotonic solution. The cell is the hypertonic solution, and water is flowing into the cell. So let's look at what this does to cells, because this is a common way that we use this information is we talk about what it's doing to the cell. So when the descriptions that we have right here, when it says hypotonic solution, isotonic, hypertonic, this is the surrounding solution that I've put the cell in. So if I've put the cell in a hypotonic solution, again, I've got low solute, high water compared to the cell, right? So the cell has less water in it because it's hypertonic if the solution is hypotonic. Water's moving into the cell. In an animal cell, that cell can go through lysis or burst because it has no cell wall. In a plant cell, you'll see animal cell, plant cell over here on the right. In a plant cell, the water moves into that cell. We talked about that central vacuole before. So the water moves into that cell. It gets stored there in that central vacuole. The central vacuole puts a lot of pressure on the cell wall. That plant cell is considered turgid and healthy. Okay? That is a turgid and healthy cell. That's where you want the cell to be. Okay? Is at that level. In an isotonic solution, that's ideal for an animal cell. So hypotonic solutions are ideal for plant cells. Isotonic are ideal for animal cells. Concentration is the same of the solute on both sides, which means the concentration of water would be the same on both sides of the membrane. Water moves in and out of the cell at the same rate, and the animal cell is normal. The plant cell will be flaccid, which means the plant is going to look like it's starting to wilt, right? It's going to start to slump. It's going to start to lean. In a hypertonic solution, so if the solution is hypertonic, that means I've got low water outside the cell. I've got high water inside the cell because the solution has lots of solute in it. And water's leaving the cell, and so the animal cell will shrivel or it will crenate, which can also damage the cell. And in a plant cell, it can go through plasmolysis. Plasmolyzed, right? Again, we're talking about lysis, bursting. In a plant cell, the cell membrane is attached to the cell wall, which you can see here. The cell membrane is attached to the cell wall. In this final picture here, the cell membrane is being ripped off of the cell wall. So that will end up killing the plant cell. That's when the plant cell, the soil is so dry, the plant is losing water out to the soil. And eventually that's how not watering your plants can kill it, because the cell membrane will pull off of the cell wall. Okay, so let's do a quick check on your understanding of osmosis. Pause it for a second. See if you can answer what's going to happen to the water, what's going to happen to the cell, which way things will move, and then we'll talk about your answer. Okay, so hopefully you paused and you tried to answer it yourself. So let's look at the cell compared to the beaker. The cell is the concentration of stuff in the cell is at 0.05, and the concentration of stuff in the environment in the beaker is at 0.03. So this one has a higher solute, which means it is hypertonic, which means I've got less water here. The environment is hypotonic, so a lower solute, which means it's going to have more water. Water flows from high to low, so this arrow kind of gave it away for you. Okay, but water should be going into the cell. So water should be moving into the cell. So the cell is hypertonic, the beaker is hypotonic, and water should be going into the cell. And we'll do lots of examples in class with this. Okay, we're going to look at one last type of diffusion, and then we'll be all set for today. So again, you hopefully heard the word diffusion, and your brain immediately needed to go to the fact that it was going to be moving high to low concentration. It was going to be passive, so no energy. And then what makes this one unique is that it requires a protein. Okay, so it requires help. Even though it requires a protein, that does not mean that it requires energy. It just requires a protein to help it get across. What this does is this allows for fast movement. For example, while we said water can diffuse across the membrane, it's a pretty slow situation. So if I have one of these channel proteins here for water, that channel protein can allow water to quickly flow through the membrane because, like we mentioned before, the sides of the protein protect the water from this non-polar region of the membrane. So I'm still flowing from high to low. I'm still passive, requiring no energy. It just requires, again, the help of this protein that's going to act as a tunnel, and it's going to be molecule specific. So not just any molecule can go through this channel. It will be molecule specific, and it will allow things to quickly move through. An example of which, like I said, would be water through what we call an aquaporin channel, right? An aquaporin channel is going to be a protein specifically that allows water to quickly flow through. So still a type of diffusion, just needs a little bit of help. Okay, think about this if you're like floating down a river, right? You've got people that can float down the river, and I go with the current. So I go from high to low. It doesn't require any energy for me to just float down the river. But then you have some people that sink like a stone to the ground, right? And they can't float. Well, those people need a raft. As soon as they have a raft or an inner tube, they're floating down the river too, and it doesn't require any energy for them. They just need a little bit of help. Okay, so next time we'll talk about active transport and look at how that's a little bit different. So like always, put in your questions into your notes, circle things that you need help with, and we'll get those cleared up when you come into class.